So here I am, it's October 31st, 2016, Halloween. It's 5:15 p.m. I'm in New Mexico. I'm here to demonstrate rhizome collection, how I've been doing it anyway, for wild hops, the Wild Hops Project. I'm here. Some ducks over there, or mallards or whatever. Anyway, so here are the two wild hop plants that I've found here. One of them is kind of all up in there. You can see kind of clustered at the top, a bunch of flowers. But what I'm interested in is down there. And there are also some vines over here. This is a little hiking trail. And on the other side there are also some vines. You can kind of see them. Get a better look there. Climbing up into this tree. Hard to tell if it's the same plant, it's the same individual or two different ones, because they're so close together. There's this wide area in between, where there's a bunch of vines over there, nothing, and a bunch of vines. Maybe they just don't come up here because it's all packed down and walked on. Maybe they're actually two different plants. I don't really know. I'm going to collect both of them. But I'm going to do a demonstration of one of them. So start here, and this one's really making it easy for me. So what I'm seeing here are little buds coming off of the stem, which is above ground. So I would imagine this is actually a legit Propagule. So for this one, I might just take one of these and not even bother digging. So maybe see if you can find one over here. Trace the vine. Wow, this is a big, thick vine. So this is actually, these are kind of emerging from here, and these are some of the biggest, fattest vines I've actually seen on this whole trip, which is kind of cool. But I'll try to find a place where they're maybe emerging from the ground and try to dig those out. So in the meantime, I've actually visited this site before and taken field notes, but I would note things such as proximity to this lake. You might even note that there are a bunch of frogs here. I would note this vegetation. These are cattails. I don't know the Latin name, but you can look that up. These appear to be willows, salix. I don't know the particular species. Still learning the local flora, but I would note that there are willows here, salix. I know the genus of this is ribes. I would note that. This appears to be a box elder, although it looks kind of weird. Usually the leaves are they're opposite because it's a maple. It's an acer. Usually the leaves, when I see them, are like this, where they have three. Each leaf has 3 leaflets, and actually this one, you can see where 2 of them fell off, so there are actually 5. And most of these looks like have 5. This one, it looks like this one, but then two fell off here. So anyway, I would note vegetation. You know, this is a different one, I think. Actually, not sure about this one. This is clematis. So things like that. I would note that the ground they're growing on is fairly flat. I would note the proximity to this trail here. The proximity to that rock face. I would note that it's pretty shaded. I mean, this looks close to full shade. I mean, considering they're growing underneath the canopy of these willows. So I would note that. You know, I would note the GPS coordinates. So I'll pull out my handy Garmin here. And I'll unlock the screen. So here's the home display. Let's see if we can get it to where it's not reflecting. Anyway, it looks something like this. So I would hit this little button, type in the name, you know, whatever the sample name is, hop, whatever, check, and then I would say save. Can enter notes here, and here's the GPS coordinates. So I'm going to cancel this because I've already marked this. What else would I note? When I start digging these, I'll have a better idea of what the substrate is like, so I'll also note, take notes about the soil. I would note, you know, just some general notes about the size of the plant, you know, in terms of approximate number of binds. Is it like less than 10? Is it about 10? Are there tens of binds? Is it around 100? You know, are there hundreds? In this case, it looks like there are tens of binds, and it's, you know, it's just sort of a ballpark figure. I would note how high it's climbing. Looks like about 4 meters or so up into the tree. I would take notes about the leaves. You know, what state are they in currently? Or they look to be kind of yellow and dry at this point. And any signs of herbivory, like you can see in this one. Some little signs of herbivory there. The camera isn't really focusing, but then I would take notes about the flowers. Most of them are way up there, but you know, again, are there less than 10 flowers? Are there tens, hundreds? You know, there's more up there. Are there seeds? You know, what density is it? Just like one seed per flower? You know, and these is kind of hard to reach. It might be helpful to have a little sort of a pruning device or something to kind of reach the higher cones, but there's a few down here, so we can kind of see if there are seeds. It does look, I wish this would focus. Kind of break some off there, so usually doing this two-handed. Oops. These down here, I'm not really seeing any seeds. And this does not surprise me because these are the only 2 plants that I've seen at this whole site. Or 1 or 2 plants, and they're both female. So that would be sort of another thing to take note of, is are there seeds or not? And I would be taking notes of all this in this little field notebook I have. So if we take a look, I haven't done the notes for this one yet, but we can take a look at another one. You know, I've done Here's one in where I did a general site description, you know, location, county, and all that. Kind of some information about the site, you know, if there's a stream or a lake or whatever, and a slope, aspect, things like that. So there's not much aspect here because it's fairly flat, but you might want to notice the aspect of that rock right there. And there are other various notes here, that things that I noticed about, you know, man-made structures, cattle fences, prominent vegetation. I kind of went over that. Density of plants. So here I would say either 1 or 2 at this whole site. And then you can see here the sort of the individual plants that I've sampled. So I have the name of it, which corresponds to, so TM, my initials, 2. It's notebook 2. It's my second field notebook. Then dash 39, which is the page number. And then AB, because I've taken, for this particular one, I took a rhizome sample and a seed sample. So again, this is for a different site. This is not for here. I'm just showing you an example. So again, I named this TM2-39AB. Again, this is a system that I found that works for me. You can come up with your own system. And, you know, GPS coordinates based on the Garmin, shade, you know, direction from stream and how far away it is, the kind of substrate it's on, if there's any slope, you know, if it's height above the water, any other topological features, you know, there's apparently trash at this site, so I noted that, you know, signs of sort of humans being there. Other prominent vegetation, you know, there were other hops. You know, again, I do the best I can. Some of these I don't know the full scientific name, so sometimes I'm just writing common names. It's just sort of, you know, doing the best I can. There are tens of vines, the leaves of senesced, flowers are female, there are hundreds of them, 1 to 2 seeds per cone, they're brown and dry. The substrate is talus, you know, of brown soil, things like that. And then the samples. One thing I didn't note here, but is the habit, you know, what is it climbing on, how high is it climbing, things like that. So I'm going to pause this and see if I can dig around and find a good place to dig some rhizomes.